Hi everyone. Today we will discuss about acetone butanol production. Both acetone and butanol are good organic solvents. Industrial production of acetone butanol by a microbiological process was first started in England. It is used in the automobile industry. The organism used for the acetone butanol production is the Clostridium acetobutylicum, and it is produced by anaerobic fermentation process. Here the structure of the acetone and butanol are given. Molecular formula of butanol is C4H10O and molecular formula of acetone is C3H6O. Next, we can list the uses of acetone and butanol. Acetone and butanol used in the automobile industry. Both acetone and butanol are organic solvents. Butanol is required for the synthesis of butadiene to produce synthetic rubber, and it is used in the brake fluid for antibiotic recovery as amines for gasoline additives, and also it is used as ester in protective coating industry. Acetone used in the preparation of explosives like trinitrotoluene. Acetone used as a gelatinizing agent for nitrocellulose in the manufacturing of explosives. Production of inoculum. Here we uses Clostridium acetobutylicum and Clostridium saccharoacetobutylicum as the inoculum. Clostridium acetobutylicum require corn medium. Then Clostridium saccharoacetobutylicum require molasses medium. The inoculum media consists of molasses, calcium carbonate, ammonium sulfate or ammonium phosphate, and constip liquor. Certain bacteria of Clostridium species are capable of producing acetone, butanol, butyric acid, and isopropanol through fermentation of molasses, starch, sucrose, etc. The relative production of these compounds may vary. It's depend upon the strain of the bacterium and the fermentation conditions. The optimum range of pH and temperature for the growth of Clostridium acetobutylicum are 5 to 7 pH and 30 to 37 degrees Celsius temperature. For Clostridium acetobutylicum, the fermentation products are acetone and butanol. Then butanol or isopropanol or butyric acid, acetic acid can be produced by Clostridium butyricum. Clostridia are spore formers and are easily maintained as soil stocks. They are not very sensitive to the oxygen, and the prolonged storage of this leads to decrease in acetone butanol production. Then the production process. Spores of soil stock are added to the deep tubes containing corn medium with 5% corn meal in water. The tubes are incubated at 37 degrees Celsius for 20 hours. These are the steps involved in the production of inoculum. Then we can move to the preparation of the medium. Starch or sugar-rich materials are used for the preparation of the medium. The fermentation is usually carried out in corn or molasses-based medium. The molasses medium contains molasses, ammonium sulfate, calcium carbonate, and sometimes cornstarch liquor. We can also use corn medium. Corn meal is prepared by passing corn through magnetic field to remove the dust and metallic debris. Then it is followed by the degerming the corn. Corn oil is removed from sprouted germ. The degermed corn is ground to a fine powder. Acetone butanol is produced by anaerobic fermentation process. The fermenters after sterilization are gassed with carbon dioxide. Then inoculum is added first to the production fermenter, followed by the addition of medium. The inoculum requirement for the production process is about 2 to 4%. Okay. The fermenter contents are again stirred with carbon dioxide gas. The fermentation is carried out with a starting pH 5.8 to 6 and temperature 34 degrees Celsius. The incubation requires 2 to 2.5 hours. The actual process of fermentation occurs in 3 phases: phase 1, phase 2 and phase 3. Phase 1 is characterized by rapid growth of the organism and production of acetic acid and butyric acid in large amount. There is a large production of carbon dioxide and hydrogen gas. Here the pH is between 5 to 6 and the time requirement is about 13 to 17 hours. The decrease in pH is due to the formation of acetic acid and butyric acid. There is an increase in the titrable acidity. In the phase 1, enzymes are produced to convert acids to neutral solvents. In the phase 2, the titrable acidity decreases due to the conversion of acids to acetone and butanol. This is called acid break. The rate of gas formation reaches maximum after the acid break. During the third phase, there is a decrease in the solvent production. There is a slight or no increase in the pH. Absolute sterile conditions are required for good production of acetone and butanol. Contamination due to the bacteriophage and lactobacilli is a major problem in the production process. Then product yield. approximately 30% of carbohydrates get converted to neutral solvents in the fermentation process. In molasses medium, the ratio of butanol and acetone is 7:3. In corn medium, the ratio of butanol and acetone is 6:3. Otherwise, we can say in corn medium, the ratio of butanol, acetone and ethanol are 6:3:1. In molasses medium, the ratio of butanol, acetone and ethanol are 6:5:3. The acetone-butanol fermentation yield several other products in addition to the gases. It include isopropanol, formic acid, acetic acid, butyric acid, etc. Acetone and butanol are recovered through continuous distillation process and fractionation.